Over the course of the last 4 lectures, we've looked in detail at how DNA satisfies these 2 key requirements for information-bearing molecules that we've been talking about. Its ability to be accurately copied and its ability to encode information about the structure of proteins. In this lecture and the next, we're going to turn our attention to the specific mechanisms by which the information that's latent in the DNA molecule gets transformed into a useful form in the structure of proteins. Going back to the framework of the central dogma that we introduced 2 lectures ago, this transformation begins in the process of transcription, in which DNA serves as a template for RNA synthesis, and then it continues with the process of translation, in which RNA, in turn, serves as a template for protein synthesis. Let me tell you in advance of the discussion that there are myriad details to both processes that we could talk about. At this point in time, a half century after the discovery of the DNA double helix, legions of molecular biologists have devoted their careers to understanding the particulars. What I'd like to do in this lecture and the next, however, is to provide an overview of these processes. We'll ignore some of the details to draw this bigger picture. So I want you, too, to focus on the main features as well, keeping in mind the broader question of how genetic information flows from DNA to proteins in cells. It's these main features, not the details, that will be useful to us as we try to understand information in evolution and biology, the organizing theme for this part of the course. So, let's begin today with transcription. Remember that transcription is a kind of DNA-directed RNA synthesis, and thus it's how information in DNA somehow passes to RNA. We call this process transcription because the biochemical language of RNA as a nucleic acid is the same as the biochemical language of DNA, a sequence of nucleotides. So we say that the information in DNA is transcribed into an RNA form. The functional role of RNA in this whole process, remember, is to act as an intermediate only, as a messenger, getting the information eventually from DNA and then on to proteins through the process of translation, which we'll discuss next time. Actually, because DNA and RNA are both kinds of nucleic acids, the fundamental features of transcription are very similar to what we've discussed previously in Lecture 7 when we talked about DNA replication. There's a key enzyme, as you might guess, that acts to synthesize the newly minted strand of RNA. This enzyme is called RNA polymerase. Not surprising, since it's a polymerase that works to make more RNA. RNA works in polymerase works in a fashion that's quite similar to the way that DNA polymerase works in the process of replication that we've looked at earlier. RNA polymerase depends, and this is the important point, on the complementary base pairing of nucleotides to bring bases into the proper order along the sequence. Remember, A pairs with U if we're talking about RNA because we have uracil instead of thymine in RNA molecules, and guanine G pairs with adenine, pairs with cytosine C. The enzyme RNA polymerase catalyzes the bond that will connect a new nucleotide into the growing strand of the RNA polymer. Okay, let's look at the process just a little bit. Just as was the case for replication, the process of transcription first requires the DNA double helix to unwind. Once the helix is unwound some, then RNA polymerase can come along, and just as we saw for DNA polymerase, it adds bases that have lined up with complementary bases on the DNA template that it's working with. This makes sense that it should be so similar to replication because both of these processes depend for the transfer of information on this complementary base pairing. Now, there are some interesting differences that we need to mention between transcription and replication. The first difference, actually, is that RNA polymerase, as an enzyme, requires a smaller cast of, let's call them, supporting characters, other enzymes that help with the job, as compared to DNA polymerase. For example, when we discussed replication, we saw that DNA polymerase requires a separate enzyme, an enzyme we called helicase, to unwind the DNA double helix before DNA polymerase could even begin its job. By contrast, RNA polymerase, at least in many organisms, can open up the DNA double helix itself. It doesn't need help doing this. A second very important difference is that only one of the two DNA strands is transcribed by RNA polymerase. Remember, when we looked at replication, both of the strands, after the helix had opened up, would be independently replicated by different DNA polymerases. But in transcription, we only have one strand copied. This makes sense, because what we're looking for is to pass on a code for building proteins, and that's going to be a sequence of the bases along the polymer, along the nucleic acid polymer. And so we only need to have one side copied, one side transcribed, because the other side would just be the same information. Although, interestingly, it would be the complementary version of the information. And we're going to come back to that point in a second. So when RNA polymerase comes in and acts, it sits into the open strand of DNA, moves down that strand, adding bases. The strand that RNA polymerase works with, we call the template strand. This is the strand that holds the information that we're interested in. As RNA polymerase moves down this strand, it follows an opening that emerges as the helix unwinds in front of it, synthesizing a single molecule of RNA, which essentially peels off from the transcription process and moves away from this. And behind, in the wake of this process, the DNA helix then re-anneals, winds back up. Actually, only a relatively small section of DNA is unwound at any one time in the process of transcription. Only a small sequence, a small number of base pairs come apart. Now, the difference between the template strand and the other strand, which we'll call the complementary strand, is kind of interesting. First of all, you might think, well, then in an entire molecule of DNA, you might ask, is one side always the side that holds the code? In other words, all of the genes that we're interested in are going to be on one side, whereas the other side is just the complement that is used to facilitate replication. And this isn't the case. If you look along the length of a DNA polymer, along a chromosome, let's say, what you find is that for some genes, the template strand will be on one of the two DNA strands, whereas for other genes, it'll be on the opposite strand. For any particular section of DNA, however, only one of the two strands will be the template strand. In other words, actually, there are a couple of interesting exceptions to this, but just a few. But in other words, it's almost never the case that you would have a template strand on one side for one gene and then a template strand that was complementary to that in some sense on the other side for a completely different gene. Why not? Well, we need a specific sequence of bases to code for the amino acids in any particular gene. And so you can imagine that if you had two sequences that coded for two strings of amino acids, it would be quite constraining to have those sequences also serve as exact complements in complementary base pairing. But as I said, interestingly, there are just a few exceptions to that, but only a few. Okay, the third key difference that I want to mention between transcription and replication is the fact that transcription starts and stops in particular places on a DNA molecule, so that only selected pieces of the DNA are transcribed, not the whole molecule. Once replication begins and we're making more DNA, at the end point of replication, the entire DNA molecule has been replicated. The whole thing is copied. But for transcription, we only need a short sequence. That makes sense. The sequence will be whatever contains the information, the gene, for the protein that we're interested in. There are specific sequences of nucleotide base pairs along the stretch of the DNA molecule that serve as markers for RNA polymerase as to where it should begin transcribing for particular genes. In general, we call those sequences of base pairs promoters because they promote transcription. For every gene along a DNA double helix, there is a separate promoter telling RNA polymerase this is where you should begin transcribing. These promoters are, as I said, particular sequences of base pairs. Promoters for different genes often have related sequences of base pairs. In other words, you can look at sequences and recognize them as related promoters because they're quite similar in terms of the sequence. But they're not exactly the same, and promoters can also have quite different sequences. The point of all this is that not only does the DNA strand contain information about the proteins that it's coding for, but it also contains another kind of information, a kind of information that is giving instructions to RNA polymerase as to where the beginning of a gene is, because we may only want to transcribe one gene. There are also termination sites, specific base pair sequences that mark where transcription will end. Taken together, the promoter and what we might call the coding region and the termination site along a sequence of DNA we would call a transcription unit. You know, interesting, when I first learned about this, I was confused, and maybe you are as well. I was thinking back when I was a student, but didn't I just learn that there are start and stop codons in the genetic code? Why do I need start and stop sequences in DNA? I mean, I've already got a start and a stop. It's a very different kind of start and stop. And remember that those start and stop codons are going to be information that we're going to use later in the process of translation because they tell the translation machinery that's building proteins where to start reading a frame and where to stop. But RNA polymerase doesn't care about those start and stop codons. It just wants to make copies, complementary base pairing copies, and so it needs separately some sort of information about where on the DNA double helix it should be doing its work. Here's another functionally important difference between transcription and replication. During replication, DNA synthesis, just one copy of the DNA molecule is made. This makes sense because, let's say, a cell is dividing. You really only need one additional copy so that each of the daughter cells can get the complete set of information. In transcription, however, a single gene, a single stretch of the DNA may be copied hundreds or even thousands of times at once. And this is because the cell may need to make a large amount of whatever particular protein that gene is coding for. And so many transcripts of RNA will have to be produced. Here's one more point I want to make, and this goes back to what we said just a moment ago about promoters. RNA polymerase, this enzyme, in many cases can't fully recognize the promoter region of a gene on its own. Now, I said that a promoter region tells RNA polymerase where to begin, but depending on the particular system, RNA polymerase might also need some information that comes from other molecules that will also bind to that promoter region. We're not going to go into those supporting players now. We'll talk about them later in the course when we talk about gene regulation, because it turns out that these other components that also recognize promoters are involved in the process by which gene transcription can be turned on and off. And just to foreshadow that discussion, the presence or absence of these other components will essentially turn on and off the ability of RNA polymerase to either recognize or somehow begin to work with a stretch of DNA. Okay, now we've seen that the role of messenger RNA is to act as an intermediate. And that seems like it's pretty straightforward. It suggests that what we should do is just have messenger RNA be an exact copy of the information along the corresponding stretch of DNA that's the gene for whatever protein we're interested in. But it's interestingly not always that simple. In fact, in the 1970s, several groups of researchers independently made a very unexpected discovery. They found in eukaryotic cells that the protein coding regions of transcription units contain large amounts of DNA, large numbers of base pairs that apparently have nothing to do with the protein that's being coded for by that gene. Specifically, they found evidence that the coding sequences of genes are interrupted by large stretches of non-coding DNA. This is a fundamental difference between the DNA found in, or between the process of transcription and the DNA found in prokaryotic cells versus eukaryotic cells. This interruption by non-coding regions is characteristic of eukaryotic cells and is not thought to occur in prokaryotic cells. Now, what this means is that somehow there has to be another step in this process, because RNA polymerase is going to start at the promoter of a transcription unit of a gene and just make a transcript that's continuous for that whole region. But we know now that some of the bases in that transcript are irrelevant. So there must be another step where that irrelevant information is excised from the transcript. It's not a trivial amount of information, or at least it's not a trivial number of base pairs. Let me give you some ballpark figures. The average size of a transcription unit, I mean, this is a real rough ballpark, but the average size of a transcription unit is more or less, let's say, about 8,000 bases long. After the non-coding bases are removed, if we take out the bases that don't have anything to do with the protein, then the average length of the sequence goes down all the way to only about 1,200 bases. 1,200 bases would code for a protein, say, of about 400 amino acids long, right? Because we have 3 bases for every amino acid. So that means 80 or 90% of the RNA that's transcribed on average from a typical transcription unit has to be cut out and thrown away before translation occurs. This is a huge amount of RNA to be synthesized and then simply thrown out. Well, the RNA that's spliced out, I want to give you a little bit more of an idea about the organization then of eukaryotic gene transcription units. The RNA that's spliced out isn't just occurring sort of interspersed in small pieces and in just little groups of nucleotides or all in one chunk, but instead occurs in several sort of medium-sized chunks that are dispersed across the gene. We call the pieces of RNA that are cut out and thrown away introns. This is a shortening of the name intervening segments, or it's sort of a modification of that name. We call them introns. That's the irrelevant RNA that's removed. The pieces that remain and that somehow have to be spliced back together, we call exons. These are called exons for expressed segments. So then we call the transcription unit or the product that's originally produced from a transcription unit the primary transcript, and we now know that the primary transcript of a section of DNA has to be processed by some sort of cutting and splicing, and the resulting RNA molecule we refer to as mature messenger RNA or a processed transcript. There are a number of other names that are used to describe that. There's a very, very large set of molecular machinery, protein enzymes, and other smaller helper RNA molecules that are involved in this process of RNA splicing. The first thing that has to be done, I'll just describe this briefly, is that the boundaries between introns and exons have to be recognized. And the only way to do this, you can imagine, is if there are particular sequences of bases that are characteristic of those boundaries. And indeed, there are. Particular sequences or sort of average sequences of bases will identify or could be used to identify where an intron begins or where an intron ends and an exon begins. These boundaries are recognized by special protein RNA complexes that catalyze the splicing, the cutting and splicing of these RNA transcripts. These units are referred to by the somewhat amusing name of SNRPs, which stands for small nuclear ribonucleotide protein particles, or snRNPs. What happens first is that two SNRPs find the boundaries of an intron. They identify those regions that are the boundaries of an intron at either side. And then once these SNRPs, and again, I want to remind you, these SNRPs themselves are very complicated protein RNA complexes. Once these SNRPs have identified these boundaries, they attach to them, and then these SNRPs are brought together by another set of SNRPs, and they form a fairly large complex that we call a spliceosome. So imagine that I have a string of RNA, a primary RNA transcript, and I have an intron here that needs to be removed. SNRPs will find either end of this intron, and then they will be brought together, forming a loop in the RNA transcript. And once they're brought together, this unit, called the spliceosome, will cut out the intron. They'll actually sever the RNA transcript and then re-splice the remaining ends from the exons together, and the intron is discarded. Now, there's another way, I should add one more thing for the sake of completeness. There's another way that eukaryotic RNA is modified before it goes to be translated as a protein. At the beginning of the mature messenger RNA molecule, a chemically modified compound similar to guanine is added. This is added to the 5' end, and we call it the 5' cap or the guanine cap. So every mature mRNA or processed transcript will on the 5' end always have this modified guanine. Meanwhile, at the other end of the RNA transcript, on the 3' end, a very long series of what might be several hundred adenines are added, forming what's called the poly-A tail. So we have a 5' or guanine cap on one end and a poly-A tail on the other. The point here is that for eukaryotic cells, there's an enormous amount of post-transcriptional processing that goes on. The RNA polymerase just starts the process by producing this primary transcript, and then that transcript has to be modified by having introns cut out and having these caps and tails added. You might wonder why they go through all of this. I mean, why not just have a sequence, which is the code, and be done with it? Well, it's not clear. Several hypotheses have been proposed. I just want to list these to you. I mean, I will point out at the beginning that some of these are quite conjectural and may well prove to be wrong. Some of them might be proved to be right. One possibility is that the splicing process itself, the process that removes these introns, is somehow associated with regulating the flow of information in the whole sequence from DNA to proteins. In other words, until those introns are cut out, the RNA transcript is not going to be a useful messenger to build proteins. And so perhaps introns have something to do with controlling the rate or the timing of actually producing those proteins, so that until the SNRPs forming the spliceosome come along and process that RNA, it isn't going to go on to be translated. That's one possibility. Other researchers have proposed that the introns are simply a sort of genetic hitchhiker, junk DNA, that somehow has gotten introduced into the DNA of cells and is irrelevant for proteins. Perhaps something like the action of retroviruses has just introduced DNA, and the whole process of splicing these introns out is more or less just the need for the cell to clean up the mess. That's another possibility. In some organisms, interestingly, the same transcription unit can produce different proteins, and I think this is really an interesting one, depending on which parts are cut out. So in other words, one gene's intron, or part of it, might be part of another gene's exon. So this might be an interesting way to save space on a DNA molecule. You know, you can take one large section, one transcript, and kind of stitch it together different ways to get different things. Related to this is a very intriguing observation that about exons, that in some cases, actually many cases, the exons, the individual exons of different genes are the same. I should say different genes share the same exons. The implication is that over evolutionary time, these exons, more or less intact, have been swapped between the coding regions of different genes along the length of the DNA molecule. Now, that idea also corresponds to the observation that many kinds of proteins share structural features in common. We call these structural features that proteins share in common functional domains. I'll make up a simple and oversimplified example. Imagine that you have two proteins, two enzymes that catalyze very different reactions. They catalyze reactions for the one enzyme that will have to work with compound A and compound B, and then the other enzyme will catalyze a reaction that will have to work with compound C and compound B as well. Compound B and compound C, we'll say. So both of them are going to have to interact with compound B, and we know from our discussion of protein structure and function that the way that they will interact with those compounds, their substrates, is going to involve something about the shape of a part of a nook or a cranny on that protein that will selectively bind that compound. So if we have two different proteins that are binding the same compound, they're likely to share structural features which are very likely to be to be coded for by identical or very similar stretches of base pairs along a DNA molecule. So the idea is that swapping functional units, these functional domains across proteins, might be facilitated by having smaller, easily identifiable units in the DNA, the units that correspond to exons, sort of clearly separated, so that those exons can be swapped between different parts of the DNA molecule. Remember, it's only changes in the DNA that have any evolutionary impact. So this is sort of like thinking that evolution has somehow come up with a kind of a fundamental protein toolkit that you can, or maybe sort of like a protein erector set where you want to build certain proteins, but you might always use some of the same parts to make different kinds of proteins. Those different parts might be coded for by exons that are swapped between genes. Okay, we're talking a lot about genes. We're talking more and more about genes. So I want to end this lecture by revisiting our definition of what we mean by a gene. So previously, we've referred to a gene as a stretch of DNA that somehow codes for a protein. Now we've learned that the physical basis of that code is a series of 3 nucleotide, 3 base long codons specifying a unique sequence of amino acids. Our discussion today makes it clear how this can work on a molecular level by RNA polymerase identifying those stretches and then beginning the process of pulling them out and sending them off so that they can be used to build proteins. And it also makes clear that even on a molecular level, the definition of a gene has to be more complex than just a sequence of codons coding for an amino acid series. Clearly, the gene also must somehow include the information that tells the RNA polymerase where to start and stop. And as we've seen with eukaryotic genes, the information also, or as we've seen with eukaryotic genes, the gene also will include entire stretches of DNA that are irrelevant to the functional protein. Next time, we'll come back to this process by which information goes from DNA to proteins by following the fate of the RNA transcripts that we've produced today.